Hello all, welcome to the ZOOMinar. Today's speakers are Dr. Hilal Lashuel and Antoine Loquet. First speaker is Dr. Lashuel. He received his bachelor's degree in chemistry from City University of New York and completed his PhD degree at Texas A&M and the Scripps Research Institute. He then joined the Pickover Institute for Medical Research in Long Island of New York, and then moved to Harvard Medical School as a research fellow. In 2005, Dr. Lashuel moved to Switzerland to join the Brain Mind Institute at EPFL, where he is a faculty of neuroscience and the director of Laboratory of Molecular and Chemical Biology of Neurodegeneration. Dr. Lashuel's research focuses on applying chemical biology approaches to elucidate the mechanisms of protein folding, misfolding, and aggregation and their contribution to neurodegenerative diseases. With that brief introduction, I would like to welcome Hilal Lashuel to give his talk. Get started, Hilal.
Thank you very much. Can you hear me?
Yep.
Okay, thank you very much again for the kind invitation and the opportunity to be here and share with you some of our work. Today, I would try to focus my talk mainly on our work on Huntington and some of the recent studies in our lab that is sort of forcing us to do two things. One is to rethink our understanding of mechanisms of protein aggregation and toxicity in Huntington disease. And the second one, in the need to develop new strategies and model systems to bridge the gap between our detailed molecular understanding of mechanisms of protein aggregation in cell-free system and the complexity of these processes in how they occur in the brain and in cellular system. So I'd like to start simply by introducing people to Huntington disease, and those people who do not know about Huntington, it's a neurodegenerative disorder that is caused by death of neurons in the brain. This usually manifests to a complex of set of both motor, behavioral, and cognitive symptoms. The most obvious of which is the motor system symptoms that one usually sees in Huntington patients, which have to do with difficulty in controlling movement. This is a video of our colleagues, Chris Ross, who's our neurologist, seeing one of the Huntington patients. I just want us to start by emphasizing that Huntington, again, is not a disease about motor symptoms. It has many motor, behavioral, and cognitive functions. Also, this is the main sort of the feature that the disease manifests itself and the obvious one that we see. In the case of Huntington, we actually know the cause of the disease. It's caused by mutation in the Huntington gene in chromosome 4. And it has to do in the normal individuals who have this gene. With in the normal individuals, there is a polyglutamine repeat in exon 1 of this gene that is usually less than 35 repeats. In individuals with Huntington disease, the mutation leads to an expanded version of this polyQ tract, usually with polyQ repeats in above 36 to 40. And at the pathological level, the disease is characterized, as we mentioned before, by the loss of dopamine, by the loss of neurons, brain atrophy, and the presence of proteinaceous, sort of both nuclear and cytoplasmic aggregates in neurons in the brain. So it turns out that in Huntington, the size does matter. The length of the polyQ repeat correlates very well with disease onset and disease severity. Individuals with less than 26 usually don't have the disease, and next generation are unlikely to have the disease. If you have between 27 to 35 polyQ repeats, then individual may not have the disease, but there is a possibility that the second generation could have the disease. And those between 36 to 39 are very high likely to get the disease. And the longer the repeat, as we said earlier, the more severe the disease and the earlier the onset. So it's a disease for which we know the cause, and it is a disease which is easily diagnosed by a blood test and doing a sort of a DNA test to diagnose it. So we know quite a lot about the Huntington pathology. We know that, as I mentioned, that these neurons have accumulation of nuclear and cytoplasmic inclusions, although the correlation between inclusion formation and degeneration is not so straightforward. We know that these cytoplasmic and nuclear inclusions are made of a number of Huntingtin proteins, so they're not made of a single Huntingtin protein. Biochemical analysis of these inclusions reveal that the presence of full-length Huntingtin, but also a number of Huntingtin fragments. Which of these fragments is actually responsible for triggering this process of pathology formation is not yet clear. Some of the missing links that are sort of hindering our progress toward developing diagnostics and therapies for Huntingtin include the fact that, as we said, the mechanism by which this process of aggregation and inclusion formation contributes to degeneration remains unknown. The mechanism of toxicity or the link between protein aggregation and toxicity is not so clear, and the nature of the toxic species or toxic form of Huntingtin is yet to be identified. We also know very little about the ultrastructure and biochemical composition of these inclusions and what that might, implications for this, for the disease. Therefore, understanding or sort of addressing these missing links is critical not only for understanding the mechanisms of underlying pathology of Huntingtin, but also for developing therapies that prevent these pathogenic processes associated with the disease. And this is also understanding these mechanisms and the nature of form of Huntingtin that is actually responsible for initiation and formation of this pathology is also essential for developing the right relevant animal and cellular models to study this disease. So in today's talk, I would like to talk about two main things, which is our recent studies from our lab aimed at trying to understand which forms of Huntingtin are actually responsible for inclusion formation. Is there a single form, multiple form, and does it matter? What are the implications of this? In the second part of the talk, I will highlight some of recent new insight from correlative light electron microscopy and proteomic studies that we have done aimed at dissecting the mechanism of Huntingtin formation and inclusion formation in cells. I had initially intended to talk about some of our recent work on trying to determine the cryo-EM structure of Huntingtin fibrils, but time is limited. I'll be happy to answer any questions at the end, or you can see our recent paper on this work published in JACS. Most of the work that I will be talking about has been summarized in these publications, with the exception of the first part on full-length Huntingtin, which has not been published. So Huntingtin is a big protein. It's 356 kilodalton protein. And for decades, people have been trying to determine the structure of Huntingtin without any success, mainly because in its own, Huntingtin is a very flexible protein that adopts many conformations, and one can see this in electron microscopy. So initial studies, attempts to determine the three-dimensional structure of Huntingtin were not successful until people found out that when Huntingtin is bound to another Huntingtin-associated protein, HAP40, the protein becomes stable. And this was a nice trick to stabilize the protein and determine its three-dimensional structure. And this is by cryo-EM. This is the structure of the Huntingtin protein, complex to HAP40. So we've known before the structure was solved for years that simple sequence changes, modification within the small N-terminal part of this protein called exon 1, which constitute only 3% of the size of the protein, were sufficient to reverse Huntingtin-associated pathology and degeneration. Or either increase it in the case of the increasing the polyQ repeat length in Huntingtin was shown to increase inclusion formation and degeneration, whereas post-translational modifications such as phosphorylation at serine 13 and S16 in this small part of the first 17 amino acids was also shown to be sufficient to protect against Huntingtin aggregation and toxicity in animal models expressing the full-length protein. Subsequent studies from our lab and others also showed that PTMs in this region are actually protective in different models of Huntingtin aggregation and degeneration. So the question we've had for years is, we know this, but what we didn't know is, do these changes in this small part of the protein exert their effect at the level of the full-length protein, or only in the poly-Q containing small fragments of the protein. And this has not been addressed for decades. When the cryo-EM structure was solved, we were very excited because we thought we will find the answers from the cryo structure in the context of how do modifications in this region influence the structure of the protein and its behavior. But it turns out that in the structure, this region is flexible and missing. So shown here is the cryo-EM structure of Huntington, and the gray areas reflects part of the protein that sort of represent loops or flexible domains. So we did not learn very much from the cryo-EM structure about how this region influenced the protein. It was thought then basically maybe if you solve the cryo-EM structures of Huntington with different polyglutamine length, we may be able to gain some insight. Unfortunately, the cryo-EM structure of Huntington with poly-Q length expansion of different lengths seems to be virtually identical. This is the cryo-EM structure of Huntington 17q complex to HAP40, 4060q, and 128q. When we overlay them, you see a very identical structure, suggesting so that domain remained flexible. So what we set out in the lab first to determine is to answer the question, does the full-length protein aggregate on its own? And this work was initiated by Dries, a technician in the lab, and followed by a PhD student, Enzo Moro. So what we did in this context is we generated full-length Huntington protein with poly-Q length, repeat of different lengths, 23q, 48q, and 73, so both below and above the pathogenic threshold. And when we looked at the stability of these protein and their propensity to aggregate, we looked up to 4 weeks of incubation at 37 degrees. The protein does not form any fibrils. We looked at, increasing the concentration of these proteins. We get no fibril formation at all. After 3 weeks, we see that this is the native Huntington, began to form small clumps of non-fibrillar aggregates. Because the monomeric protein is 350 kilodalton, these aggregates can actually be sedimented. But in all the conditions, including exploring different pHs, we could not find that Huntington full-length protein could make fibrillar aggregates in vitro. And what we know that for most proteins like TDP-43, for example, it has the globular domains and it has a low complexity domains which can drive the aggregation of the full length. We know if you take A beta and attach it to GFP, you can still form fibrils, the same for tau. But this aggregation domain, the N-terminus of Huntington, seems to be not sufficient to drive the fibrillization of the full length. And we argue that because of the large size of the protein, there is quite a bit of structural hindrance to form a very tightly packed amyloid fibrils. But what we know is also, if we look at the effect of these sequence changes at the level of the exon 1 fragment itself, then they do predict the aggregation properties and the toxicity of this protein in cells. We know that if we take exon 1 by itself, either in vitro or cells, the longer the poly-Q, the higher the propensity of the protein to aggregate. In our lab, we have looked at the effect of all the known PTM modification within N17. And usually the effect of these PTMs is that we see in vitro is predictive of the behavior of the protein in cells. So for example, we know that phosphorylation at T3 and phosphorylation at S13 inhibit aggregation. And we can show that, kinases that phosphorylate at these residues also inhibit aggregation cells. So this led us to suggest that perhaps the effect of the poly-Q expansion and these PTMs in the N17 is mediated primarily on the fragment form of the protein and not the full-length form of the protein. So to summarize this part, what I've shown you is that the full-length protein does not form fibrillar aggregates. The effect of some PTMs and poly-Q expansion at the level of exon 1 predict their effect on Huntington aggregation and toxicity in vivo. And this led us to hypothesize that proteolytic processing of the full-length Huntington may be required for Huntington aggregation and inclusion formation. To begin to test this hypothesis, one needs to first identify which are the natural fragments that are normally generated by Huntingtin protein in cells, and then which enzymes are usually responsible for this. Unfortunately, right now, we don't know the exact answers for this, but what we did to test this hypothesis is to try to explore, enzymes or proteases that can give us rise to fragments that are similar to those seen in vivo. So for many years, exon 1, as many of you know, has been this fragment is the most studied fragments, simply because there is strong evidence that this fragments actually occurs by naturally through alternative parent splicing, and that it also contains this poly-Q repeat, and it's present in diseased brain, and in animal models, it seems to be sufficient to reproduce many key features of the disease. In the context of in vitro studies, because of the small size of this protein, and because people developed initially efficient expression system using GST fusion proteins and others, it became the most dominantly and mainly studied fragment of Huntingtin. But we know, as I said, that Huntingtin undergoes proteolysis, and these are some of the fragments of Huntingtin that have been actually shown to occur in vivo. And our hypothesis is that Huntingtin processing here could result in the generation of these fragments, and these fragments are likely to be the main drivers of Huntingtin aggregation. So to test this, no one has looked at whether you can induce Huntingtin, the generation of exon 1 from the full-length protein and the generation of exon 1 fibrils. And in our lab, we're basically testing these two hypotheses. One is that proteolysis and generation of different N-terminal fragments is a key step for formation of pathology. And the second one, which I'm not going to talk about today, is that post-translational modification play a key role in regulating susceptibility of Huntingtin to hydrolysis. So what Enzo in the lab did is basically we went and we tried to identify proteases that could cut the full-length Huntingtin protein without any cleavages within exon 1. And also, if they cut outside exon 1, is okay. And what we found here is the criteria was it does not cleave within exon 1, and that exon 1 could result in production of N-terminal fragment of different length. And we found one of these proteases, which is RC, which cleaves right just after exon 1. It also leads the production of a number of fragments at 128 and 167, which are similar to Huntingtin fragments that have been observed in vivo. And therefore, we asked the question, if we take full-length Huntingtin protein, treat it with this enzyme, it will generate these fragments. And then we asked, is this be sufficient? Will we get aggregation of these fragments? And which fragments will lead to the formation of Huntingtin fibrils? This is just an experiment using a wild-type full-length Huntingtin, where you can monitor the cleavage of Huntingtin by RC over time. And what you could see is that quickly within the first hour, we see the generation of a number of fragments that becomes later dominated by these N-terminal fragments. And you see the gradual buildup of exon 1 after 1 hour. And these fragments decrease over time. So when you monitor this reaction by electron microscopy, this is what you see. Of course, in the beginning, you have a mixture of different fragments. So early on, you don't have the generation of these N-terminal fragments. You have mostly globular structures. At time 4 hour, where you have a mixture of both N-terminal fragment and exon 1, we began to see this non-fibrillar aggregate, we believe are composed of longer N-terminal fragments. And if you wait longer after exon 1 is formed, then wait 24 hours, then you can find the formation of fibrils. This is the first time that exon 1 fibrils are generated through processing of the full-length Huntingtin protein. Therefore, we can establish this proteolysis as a potential mechanism for generation of Huntingtin aggregates. Now, as I mentioned, there are different number of fragments that have been observed for Huntingtin in vivo, but the aggregation properties of these fragments have not been investigated because they're very difficult to express and purify and handle. So Dries, Raja, and Iman in the lab set out to explore the mechanism of aggregation of these different fragments in vitro. And I'm here summarizing this result. This work is summarized, is presented, this Jack's paper we published last year. And what you could see is that when you have exon 1 of the Huntingtin protein, it aggregates very rapidly in vitro and forms mainly these single short fibrillar structures. If we go to a longer fragment of Huntingtin, we extend this by about 15 residues to Huntingtin 104. It does not affect dramatically the kinetics or the morphology of Huntingtin aggregates. Now, if you start to go to longer fragments, 140 and 171, where you have this, part of a globular helical repeat domain, now we see a dramatic change in the type of aggregates that are formed. We see instead of these single filament, we see these bundles of aggregates that tend to pack together. So what Raja and Iman did is they set out to dissect the mechanism of how these large aggregates form. And what they showed basically is that at very early time point, these longer N-terminal fragment Huntingtin 71s self-assemble into these globular structures, and then goes on to form these very diffuse structures where you can see the proteins are beginning even to line up to form fibrils, but they don't form smooth fibrils. However, over time, what you see is that emanating from these sort of amorphous type of or less ordered structures are fibrils that emerge and grow sort of on the surface of these aggregates, which then have a very high tendency to self-associate. And when we try to understand the mechanism of this, we believe that this initial part could be driven by sort of a phase separation related event. And the mechanism the model we proposed in the case of the longer fragment, that the aggregation is actually driven by this globular domain and not by this poly-Q or N17 domain. And this leads to the clustering of Huntingtin, which then the poly-Q and NC17 undergo conformational change and begin to self-assemble at the end of these, giving to these emanating fibril structures. So the idea here is that this process is initiated by the globular domain rather than the poly-Q domain. And what is nice is that Iman has done some cryo-electron microscopy, and when you look at the fibrils under cryo condition formed by this 171, you can see nicely the filament here decorated by these individual globular domain representing this helical repeat domain that we see there. And these are some more close-up images of this protein. Now, we had previously described that in this region of 128 to 138 is a very highly amylogenic sequence that induces the aggregation of Huntingtins in addition to the poly-Q. So we reasoned if this is really the mechanism of driving aggregation, then removing this hydrophobic domain should prevent the clumping or its lateral association of these aggregates or disfavor this aggregation mechanism. And in here you see this is a exon 171 with 128 to 138 removed. And you can see we began now to go back to forming these individual filaments. The second experiment to test this hypothesis was if we add simply this globular domain of 105 to 171, we should be able to compete for these interactions and prevent the aggregation of this protein. And what you see here is a dose-dependent inhibition of the aggregation of 171 by this globular domain 105 to 171. So establishing that this mechanism for these longer repeats likely holds. Now we tried to look at the aggregation of other longer repeats, 586, for example, and when we study these different fragments in cell, we could never in our hand get this 586 larger N-terminal fragment to form inclusions, especially nuclear inclusions in neurons. And we tried to look at this, so we expressed the protein in vitro. We studied its aggregation at different pHs, at different incubation, 37 degrees. We could never get the protein to aggregate. It turns out that if we play with the phosphorylation state of this protein by inducing hyperphosphorylation at 3 to 4 sites, that the protein can begin to aggregate. And now we can see the formation of these protofilaments and fibril-like structures. And in some conditions, we can see the formation of these single and twisted helical filament by this full-length protein. So why I'm saying this is, to summarize this part, what I'm trying to show you here is that the mechanism, not only the kinetics of aggregation is different for the different fragments, but also the mechanism of aggregation is different. Which part of the protein is responsible for initiating the process is dependent on the N-terminal length. And for example, in the case of exon 1, we have two different proposed models now, one where aggregation is driven by N17 or the aggregation of the poly-Q domain. Whereas I've shown you in the case of longer repeat, this process could be initiated by this globular domain to bring all these exon 1 into place, and then fibril formation could be initiated on the surface of these aggregates. I've also shown you that in the case of the 586 fragment, the aggregation is initiated by a post-translational modification of the protein. So to summarize this part, what I hope I've been able to convince you is that the sequence outside the poly-Q domain could play an important role not only in the initiation of aggregation, but also in determining the aggregation pathway and the final morphological and structural properties of the aggregates. The different N-terminal aggregates could have distinct aggregation mechanisms, and that the single poly, what this means is that a single poly-Q targeting anti-aggregation strategies may not be effective to inhibit the aggregation of different fragments. And that if we want to target Huntington aggregation in vivo, then we will need to take into consideration the presence of multiple fragments, and perhaps future screens should include multiple fragments, be done against multiple fragments of Huntington, not one. And finally, we still don't understand when these different fragments that form, how do they interact with each other and influence the mechanism of Huntington aggregation. Now, I'd like in the last few minutes to talk about our effort to try to understand how Huntington aggregates form in cells. And we know from the ultrastructure studies of Huntington aggregates that they look like these. And what you could see here is that there is the Huntington aggregates that appear to be fibrillar, but they're surrounded by different cellular sort of structures such as mitochondria, organelles, and lipids. And so our goal is, in vitro, this is what Huntington aggregates look like. As I showed you, they're mostly fibrillar. This is what Huntington aggregates look in the brain. And what we're trying to do is to figure out a way where we can develop model systems that reconstitute the complexity of Huntington aggregates in the brain. And that's the idea of trying to develop cellular model to do this. So to do this, what we did in the lab is we tried to study the mechanism of Huntington aggregation. As I said, Huntington aggregates form in the cytosol and the nuclear. So to do this, we use different model systems. We use hex cells because overexpression of Huntington and hex cells result in predominantly cytoplasmic inclusion. So we can use it to study both cytoplasmic and nuclear. In neuronal cells, the overexpression of Huntington results predominantly in nuclear inclusion. And some of you may be familiar with this work that we have done in our lab in the case of Parkinson's disease, where we have recently developed and published a model where we can reproduce the complexity in Lewy bodies in the brain in primary neurons. And what we use here to characterize these is a technique known as correlated electron microscopy. So we use condition to introduce synuclein aggregation in neurons. We monitor how these aggregates evolve over time. And then we use electron microscopy to see how these aggregates actually evolve over time and how they interact with other cellular components and lead to inclusion. So we extended the same approach to look at Huntington. And in hex cells, if you overexpress Huntington, in this case, we're working with exon 1. And I would like to emphasize here that we're working with exon 1 in the absence of a GFP tag. And I'll emphasize why that's important later. When you overexpress Huntington 1 in cells, you see always the formation of these ring-like structures. And we tried, although everyone has reported it, nobody understood why we have these ring-like structures. We tried different antibodies, and irrespective of the antibody, we get the same ring-like structure. And this led us to suggest that either these aggregates are so compact that the antibodies cannot get into the middle, or that there is something else in the middle and Huntington is mainly organized in the periphery. Now, when we look by electron microscopy, and this is all work led by Netta Ridwan, PhD students in the lab, now we can see the detailed ultrastructure features of these Huntingtin inclusions. And what you can see is that they are not simply made of aggregates. The center of these Huntingtin inclusion we show are made of dense accumulation of fibrils in complex with lipids, as well as ER membranes. And at the edge, you can see radiating filaments of Huntingtin fibrils. So this ultrastructure organization clearly explains this sort of immunohistochemistry pattern we see in cells. So then we basically, you can see this is a 3D reconstruction of these inclusions that allows you to see how do these aggregates interact with the cellular compartments around them. And you can see that the formation of these aggregates are surrounded by these ER structures. So there is an mitochondria in the vicinity. And so these inclusions are not only made of fibrils, there is hundreds and hundreds of proteins that are there representing different cellular pathways and cellular compartments that are there. So this shows that similar to what we see in Parkinson, the process of inclusion formation is distinct from that of aggregation. So there is Huntingtin aggregation, then Huntingtin inclusion formation, which appears to be a much more complex pathway. We tried to do functional studies to see how these inclusions influence the cellular function. We did see an increase in ER exit site remodeling, which suggests that this process could contribute to ER Golgi secretory defect. In the case of mitochondrial function, we see an increase in mitochondria fragmentation and mitochondria respiration, which we believe represent an adaptive mechanism to generate sufficient energy to cope with this unfolding response. In the case of these hex cells, we actually don't see quite a lot of cell death. So the formation of these inclusion may either degenerate, cell death take long time, or that they could actually, constitute an adaptive response. We tried to look at the effect of the polyQ length and the ultrastructure properties of the inclusions, and we do see a big effect. You see, this is a 39Q. We form mainly compact inclusion that are similar to the core. We don't have these radiating periphery. It could also represent an early stage where this is a late, secondary phase of growth in the case of the 72Q. Interestingly, this N17 region was thought to play an important role in membrane interaction. Therefore, we thought if we remove this, it could alter the ultrastructure properties of the aggregates, but actually what we saw is that when we remove the N17, it had no effect on the ultrastructure properties of these, and this sort of core and shell structure is still there. And so since these cells form both cytoplasmic and nuclear inclusion, we also looked at the structural properties of the inclusions. And what you could see is that these nuclear inclusions are completely distinct from those ring-like cytoplasmic inclusions. They seem to be made of purely dense accumulation of fibrillar aggregates. We don't see any sequesterization of membrane structures, suggesting that the mechanism of Huntingtin inclusion is in the nucleus could be different from the cytoplasm, and therefore the mechanisms of toxicities associated with each could be different. This is just a large image of these cytoplasmic and nuclear inclusions. So what we think is happening in terms of in cells is that you have the aggregation of mutant Huntingtin, which form these dense accumulation that constitute of fibrils that represent the core of the structure. And then these aggregates began to grow on the periphery through growth of the fibrils by monomer addition. They began to interact, they're much more loosely, and they're able to interact with proteins and membranous structures, leading to the formation of these complex inclusions that we see. We believe that this first phase is most likely driven by phase separation, because as you can see here, it's a very fast process. It's a process, if we transfect these cells and then nothing happened up to 24 hours, and then it's a very fast where you see nearly complete depletion of all soluble protein when the inclusions here, form. And what the implications of this is that one thing I wanted to say is that if you use a GFP protein this process does not occur. And I'm not going to explain that a lot. Let's talk about this a lot, but what we notice is that if you use GFP fused, Huntingtin fused to GFP, then the type of aggregates you form are mostly fibrillar in nature, resembling those we see in vitro, and do not reconstitute the complexity of inclusion formed by the naked protein. In the Nature Communication paper, we have detailed comparison of proteomics and toxicity studies, and we can show that these have distinct interactome, proteome composition, and toxicity. Therefore, when you use GFP fused to Huntingtin, you're able to reproduce Huntingtin fibril formation, but not Huntingtin inclusion formation. Okay. The implications of our work is that most effort have focused initially in trying to target the early pathway, which is to block the aggregation of Huntingtin. And what this work and the interaction with these proteins and cellular sequestration of a lot of functional protein suggests that this growth phase of the nucleation and that nucleation inclusion maturation may constitute another important step that can be targeted. So what about nuclear inclusion? And I'll finish with these couple slides. We tried to study the structure of the nuclear inclusions. We did this in primary neurons. If we express exon 1 in primary neurons, we can see the formation of inclusions over time. The size of these inclusions tend to increase over time. And basically, you can see that this is more large inclusions forming at day 7 and day 14. And in this case, we see a direct correlation between inclusion formation and toxicity. We see an increased toxicity in neurons under condition where these inclusions are formed. So we tried to look at this by electron microscopy. And what you could see here is basically that we can electron microscopy reveal structure that look very similar to what we see of nuclear inclusions in the Huntington brain. They appear to us very fibrillar, but very difficult to decipher. So this is why we did cell tomography with Sergei Nazarov in the lab. And when you do this tomography, you begin to see the fibril architecture. And to make it easy for you to see, this is what Huntingtin nuclear aggregates look like. You can see that they're made primarily of fibrillar aggregated form of Huntingtin. And with this, I'd like to sort of end by emphasizing sort of the main messages from the cellular work, that is cytoplasmic Huntingtin inclusions are composed of complex mixtures of aggregated mutant Huntingtin, different cellular proteins and membrane organelles, including members of the endosomal system. That nuclear and cytoplasmic inclusion exhibit distinct biochemical composition and ultrastructural properties, suggesting different mechanism of aggregation, inclusion formation, and toxicity, and that these differences in the cellular environment and interactome could influence the mechanism of aggregation and possibly influence their contribution. So we need further, we viewed aggregation of the context of only the formation of fibrils. We need further studies to investigate the role of lipids, organelles, and Huntingtin interactome prey in the formation of Huntingtin aggregates and toxicity. We are planning to extend this model to study longer repeat. And there is a hypothesis that was first suggested by Steve Finkenbeiner, suggesting that cytoplasmic and nuclear aggregates could actually serve different mechanisms, where the cytoplasmic aggregates may have more of a protective adaptive response, whereas the nuclear aggregates could be more toxic. With this, I'd like to finish by thanking you and acknowledging the people who've done the work. I've sort of mentioned their names. So a lot of the work on the N-terminal fragment was done by Raja and Iman and Anas, Nathan with the help of Anlor and other postdocs in the lab, Niran, Johannes did a lot of the work on the cellular models. Dries and Enzo from the lab did all the work with the full-length Huntingtin, and we're grateful for all the help from the bioimaging facility and our collaborators in looking at functional effects of Huntingtin aggregation and ER stress, Hisso, Farhan and his lab, and thank you for your attention.
Thank you very much for a great talk, Hilal. Let me quickly go through the questions in the Q&A folder. There are 2 questions already. From Goming, what are the relative populations of different fragments in cells? Will that vary from patient to patient?
Yeah, actually, this is a fantastic question, and the answer is we don't know. We're actually, started a project with Richard Fall in New Zealand because he has a very nice collection of brain to remap the distribution of Huntington species in different brain regions. And this has not been done. This is a big problem, We focused on exon 1 because of the alternative splicing hypothesis and because it's easy to make. But we know that, for example, 128, 171, 586, all these fragments exist. The relative distribution of these in the pathology is not known. The relative distribution in different brain regions is not known. We hypothesize that relative distribution could actually partially explain the clinical heterogeneity of the disease. So the answer is we don't know, and we need to know, because it's only through knowing that we can develop the right models to study this disease.
Okay, so connected question is, will a longer fragment be transformed to a shorter one by protease during the fibrillation process?
Yes. So the answer in our model system, where we start from full-length protein, we see sequential fragmentation, even leading to mostly exon 1. Now, one thing that could complicate this is in this experiment, when we actually look at soluble and insoluble distribution of Huntingtin over time, the longer fragments seem to sediment faster than exon 1. In other words, if you form a longer fragment with higher propensity to aggregate, that process could limit the further processing to generate exon 1.
Great, thank you. Next question from Martin Mursal. Do you have some insight why the globular domain would promote formation of mesoscopic phases instead of small micelle-like aggregates you showed?
It's a very good question, but we don't know the answer. We're trying to do more mechanistic work on this. But as I said before, when we studied the sequences within these repeats, we identified multiple stretches that actually between 110 and 140, there are 8 to 9 amino acid stretches that if you synthesize these peptides by themselves, they're very highly aggregation prone. So the idea is that if these fragments are generated, then these domains are somehow exposed. They're as sticky as the polyQ domain and would be sufficient to drive. The fact that they don't form fibrils or drive fibril formation directly could be because they're only partially unfolded, that the rest of the protein is still maintain some helicity.
Connected question to that is, do you assume it's micelle aggregation mediated by the polyQ domain that eventually allows the polyQ domain to take over and promote fibril formation?
We think that's one of the models we're testing, yes.
Okay. And the last question is from an anonymous attendee. Does the fibrillar twist depends on fragment length? Do you see fibrillar twist for short fragments?
Yeah, I didn't have a chance to present this work, but you can see it in our JAX paper. We have done the cryo-EM structure of the exon 1, and the exon. So far none of these fragments seems to form a helical filaments. Most of the Huntington internal fragments do not form twisted helical. And in the paper, we explain why, because there is quite a lot of out-of register movement and intra and interfibril heterogeneity in terms of the length of the polyQ strands and the helical and the turns. So we've only seen the formation of helical filaments with the 586 after we induce phosphorylation. And that's the reason we have not seen a cryo-EM structure of any polyglutamine fragments. Because of their ability to go off register, you don't get that twist.
Great. Thank you very much for the great talk and the great discussion. So if we can stay around towards the end, we'll get more questions from the participants. Let's move on to the next speaker, Dr. Antoine Loquet. from University of Bordeaux. Antoine received his PhD degree at the University of Lyon in 2009, working with Dr. Anya Brockman on developing solid-state NMR spectroscopy for protein structure determination. And then he was a postdoc between 2009 and 2013 at Max Planck Institute, Göttingen, working with Dr. Adam Lange on the solid-state NMR application to study protein assembly. Since 2014, he is the research director at CNRS and the University of Bordeaux, France. His current research focuses on the structural biology of functional amyloids by solid-state NMR spectroscopy. Antoine, it's all yours. Take over.
So thank you very much. I would like to thank you and also all the co-organizers for this wonderful Zoom seminars. And today I'm going to present 3 topics. The first one is related to the methodological development of cell-free synthesis for amyloid proteins. Second topic will be related to the molecular basis of amyloid cross-seeding. And the third topic focuses on the study of heterotypic interface between functional amyloids. But first, I would like to thank the people who contributed to this work. So let me thank all people in the group, as well as past members, especially Alonce Lenz, Azen Daskalov, and Jaya Shenoy. And I would like to thank also our co-workers, especially the group of Sven Soap, with who we collaborate since now almost 10 years on prions and functional amyloids. So let me start by very rapidly introducing functional amyloids. So we all know pathological amyloids, since they are involved in various neurodegenerative disorder, and they are often associated with misfolding processes and a loss of function mechanisms. However, the villains can also become the good ones, and amyloids can also be non-pathological, but beneficial for cells. In fact, most of amyloids are actually not pathological, but functionals. Indeed, nowadays, it's several thousands of functional amyloids that have been identified. And in 2019, we released a special issue in the Journal of Molecular Biology on Functional Amyloids, together with Sven Soap and Diego Romero. And we tried to cover a broad range of function and also microorganisms associated with these functional amyloids. So in my group, we have a strong interest in functional amyloids involved in various organisms or biological processes. And especially we are interested in amyloids involved in signaling mechanisms. So the long-term objective is to understand why the nature has optimized the amyloid fold to be so efficient in signaling, and what could be the advantages of amyloids compared to globular proteins to execute signaling mechanism.
Antoine, can you go to the presentation mode?
Sorry?
Can you go to the presentation mode?
Oh, yeah.
Yeah, good. This is good.
Sorry. Okay, so I'm going to start with methodological development we have carried out to implement the cell-free synthesis of amyloid proteins. And this is just a reminder for those who are not familiar with cell-free synthesis. It's an approach performed in vitro without the requirement of living cells. In turn, transcription and translation processes are achieved in a mixture composed of cell extract, supplemented with amino acids, DNA, energy regenerating system, and so on to achieve the in vitro expression without the cell membrane physical barrier. And this approach can be very powerful for NMR studies because isotopically labeled amino acids can be added to the mixture to obtain selective labeling. So cell-free approach have been implemented for solid-state NMR study during the last decade, mostly to study membrane proteins that are especially difficult to express in E. coli. Okay, so actually, why do we want to implement and develop the cell-free synthesis of amyloid proteins? Well, that's because sample preparation for amyloid proteins is still very challenging. as an example, in my group, students spend easily 50% of their research time to perform a sample preparation. It's time consuming and it's still technically very difficult. And because the production yields are usually low, and this protein also aggregates into inclusion bodies during recombinant pattern expression. So we need to isolate them from the inclusions and also to solubilize and also to perform additional purification step. And on top of that, also some amyloid domains, especially the small amyloid domain, can also be cytotoxic for bacteria. So usually when you talk to biochemists about inclusion bodies, they will tell you that they hate inclusion bodies because it means that you need to use biochemical denaturation and also refolding to get you protein assembly. However, there are one advantage that is associated to the aggregation into inclusion bodies is that actually you can recover quite a lot of protein. I mean, in terms of quantity of protein from these inclusion bodies. So we have implemented the cell-free synthesis for amyloid proteins by adapting protocols developed and based on the dialysis between a feeding mixture and a reaction mixture. So we tested our approach using the prion-forming domain of HETES, which is a fungal prion that form amyloid fibrils, and I will come back to the biology of this system later during my presentation. So what was quite surprising for us was the almost spontaneous aggregation of the expressed protein inside the cell-free mixture. Here what you can see is an electron micrograph of HETES fibrils obtained directly inside the reaction mixture. And we use this biochemical feature, I mean the fact that the expressed protein spontaneously aggregate inside the cell-free reaction to propose a very straightforward protocol for which the pellet obtained in the reaction mixture is simply washed with water, and then we directly use this pellet to fill the solid-state NMR rotor. Basically, it means that contrary to what we usually do during a recombinant bacterial expression in E. coli, here we do not perform any biochemical denaturation, additional protein purification, or refolding to obtain the fibril sample. And we tested our approach by implementing a strategic isotope labeling on HETES. So we added unlabeled amino acid and also carbon-13 label glycine, isoleucine, and valine. Okay, glycine, isoleucine, and valine. And we combined this strategic isotope labeling with the latest solid-state NMR technology, so namely the use of fast magic angle spinning to achieve proton detection. So what you can see here are two-dimensional solid-state NMR experiments. In orange, it's an NH correlation of HETES prion fibrils express and purify from E. coli and uniformly carbon-13 label. And in blue, these are the same fibrils, but this time they were cell-free synthesized and selectively labeled on valine, isoleucine, and glycine. So comparable chemical shifts are observed and suggesting that the three-dimensional amyloid fold, I mean at the level of the local conformation, is virtually the same between the cell-free synthesized amyloids and the fibrils obtained from recombinant expression, denaturation, and in vitro aggregation. It's unexpected and I think quite remarkable, considering that the cell-free reaction mixture is complex. It's not very well controlled, and it's also not purified. So compared to the situation in the conventional protocol where we in vitro aggregate HETES from a very pure HETES monomeric solution. So we also observe a comparable spectral resolution. It also indicates that at the level of the local structural polymorphism, we have a very low structural polymorphism in cell-free synthesized HETES, which is again quite remarkable considering that the aggregation medium is also not controlled and also pretty complex. So we also tested the incorporation of fluorines, so fluorine label, and we could also demonstrate that it's also possible to perform this labeling of amyloid fibrils using this cell-free approach. So in collaboration with the group of Gilles Guichard, who recently reported the synthesis of hexafluoroleucines. We tested the cell-free synthesis of fluorinated amyloids. So HETES has 3 leucine in its amyloid core. So we could expect 6 fluorine signal, and actually we could detect 5 out of 6 fluorine signals. So this result provides a proof of concept for for cell-free synthesis of fluorinated amyloids with good yield and also with a limited structural polymorphism. One has also to consider that the incorporation of fluorinated amino acid is usually extremely challenging when you work with conventional bacterial expression systems. So that's also a big advantage of this cell-free synthesis approach. So the last test and the last experiment, and this one was also very important, was to study the functionality of these cell-free synthesized amyloids. So how synthesized HETES fibrils were able to trigger a regulated cell death reaction in their natural host, which is the fungal Podospora serina. So in terms, and to make a long story short, it indicates that the cell-free synthesized amyloids are infectious, and they also keep their prion properties. And we try to summarize our findings and to put them in perspective compared to previous report. So we observe that cell-free synthesized HETES form a canonical beta-solenoid with a limited structural polymorphism. This is what we can see from the NMR line width. And it's something that was observed from recombinant in vivo expression of the same protein in E. coli, followed by denaturation and an in vitro aggregation at pH 7. The cell-free synthesized amyloids are functional prions, and they are also infectious in their natural host. And this is a feature that was, of course, observed in vivo, but also for fibrils obtained at pH 7. And they have a different behavior compared to the HETES fibrils aggregated in a non-physiological environment at pH 2. So to some extent, we can hypothesize that the complex and the crowded environment that exists in the cell-free reaction mixture might also partially reassemble some feature of the environment for the inclusion bodies. And that's quite interesting because there has been also a report that actually HETES aggregates isolated for inclusion bodies are also infectious. So we think that it can also be a good model to study the aggregation in inclusion bodies. Good. So for the second topic, I'm going to present the investigation of the molecular basis of amyloid cross-seeding. But to start, I would like to introduce signalosomes. So these are intracellular sensors for pathogens, and they control crucial mechanism in immunity. They are subfamily of what is called the SMOC. These are for supramolecular organizing centers. And they usually share a tripartite architecture with a receptor, a downstream accessory protein, and an executioner that is usually a self-death executioner. And they have been observed in plants, in fungi, and also in animals. And in fungi, the gene architecture can be slightly more complex and involve actually 2 genes. So one has the receptor, one encode for the receptor and the downstream protein, with an amyloid domain that is attached to the enter, while the executioner is located on another gene that is adjacent, also with an amyloid domain that is attached to its C-terminal domain. And the last couple of years, we have been working together with the team of Sven Soap on the characterization of a fungal amyloid subfamily of this signalosome that are called HRAM. And in particular, we have focused on one protein that is called HELF. So HELF is a fungal prion that has the ability to trigger self-death reaction upon a templating mechanism that involve its amyloid domain. And we also know actually how to recombinantly express and also to obtain beautiful fibrils from HELF. So we have used this system and tried to see how far we can go with atomic resolution structure determination of amyloid fibrils using the latest methodology in solid-state NMR, so based on magic angle spinning, and also combined with a high-field NMR. And we demonstrated that atomic resolution information can be obtained based on a very small quantity. So it's about 200 micrograms of recombinant carbon-13 and nitrogen-50 label amyloids. And using solid-state NMR structural information, we demonstrate that high-resolution structure functional amyloids can be determined using proton-proton restraint. In analogy, I mean, it's not exactly the same methodology, but we can make an analogy to structure determination by solution NMR using proton-proton NOEs. So the health form canonical beta-solenoid structure with two distinct repeats that we call R1 and R2. So I'm not going to say too much about the structure, since we are mostly interested by the biology of the system more than the structure itself, although this was a very nice model for us to implement solid-state NMR techniques to study amyloid fibrils using proton detection. So we determined the fold of health, which is actually highly comparable to HETES. HETES is another fungal prion for which the structure was also solved in the group of Beat Myers. And health belongs to the HRAM5 family, while HETES is from the HRAM1 family. Okay. And a very interesting observation is that the two sequences only share a 17% sequence homology, while when comparing the structure, they share the same, virtually the same backbone amyloid fold. Actually, there's one part that is a bit longer for HETES. You can see in yellow. That's just because there is an unstructured loop between the two repeat that is longer in the case of HETES compared to health. So this is, we believe that this is the experimental demonstration of a very close, not to say identical, structural homology of sequence distant prion homologues, especially considering that both prions are actually from the same natural host, so from this filamentous fungus. And a very interesting experiment, and it was highly surprising for us, is the fact that HETES and health actually do not cross-seed. That's something we have tested in vivo and also in vitro. And so because the two prions are from the same organism and they share the same amyloid structure, and they are both involved in the same regulated cell death reaction, what could actually expect them to be able to cross-seed, but that was not the case. So in order to explore this aspect of sequence-to-fold conservation of the beta-solenoid fold, we have tried to investigate how far we can go with low sequence homology to still maintain a prion behavior and importantly, to still keep a structural homology. So we design a minimal prion, we call it HEAD, so for HETES distant prion, and we engineer the sequence of HEAD to keep a very low score of homology, so of only 5% compared to HETES. So of course, it's very easy to change amino acids and to go very low in sequence homology, but the point here is to change this amino acid, but to still keep the amyloid behavior and also to still keep the prion functionality. So we managed to do that, and actually HEAD, so this synthetic HEAD, also form a prion in vivo, and we perform solid-state NMR to rapidly derive its secondary structure. So it's typically here that I think solid-state NMR is a very good technique because we can obtain this information using a minimal amount of spectrometer time and also of spectral analysis. So we could show that actually, because HEAD also has the same structure, that the amyloid fold can adapt to an extreme level of amino acid substitution. In that case, we can even reach the so-called midnight zone of sequence homology, which is inferior to, I don't remember, it's 10 or 15% of sequence homology. And it's something that was demonstrated a long time ago for globular protein, that you can have structural homology even if you are in the midnight zone of sequence homology. And it's also something that we could show, at least in this example for this class of protein fold, so the amyloid fold. And next, we wanted to see if by actually increasing the sequence homology, we can now breach the seeding barrier. And for that, we engineer another chimeric prion. We call it HEC. So for hetes closer prion. So HEC is a chimeric prion. Actually, we increase the sequence identity now to 40% compared to hetes. So we change the amino acids to now reach sequence identity of 40%. And it was amazing to observe that with this engineered sequence, we could now breach the seeding barrier and actually cross-seed so HEC with hetes or with health independently. And again, we perform a solid-state NMR analysis of HEC to also show that it keeps the same structural architecture. So it suggests that actually the presence of we probably have the presence of a very subtle mechanism that is encoded at the level of the sequence, also at the level of the structure, but not only at the level of the structure, I mean, at the level of the three-dimensional backbone fold that allow or do not allow the cross-seeding between the prion entities. And that's an aspect we want to explore further in the future by engineering more sequences to try to rationalize actually the specificity of this cross-seeding between amyloids. Okay, so for the last part of my presentation, I want to focus on the investigation of heterotypic interface in amyloids. And so I go back to this beta solenoid fold that is made by the supramolecular arrangement of two repeats. These are called R1 and R2. So it's the case in health, but it's also the case in hetes. And as I mentioned earlier in my presentation, there is another amyloid domain that is also encoded by the adjacent gene. So the protein is called NWD2, and to make the story more simple, its amyloid domain is called R0, in analogy to R1 and R2 that is present on hetes. And back in 2015, we used specific isotope labeling to investigate the secondary structure of R0 fibrils, but at that time we didn't manage to get its high resolution structure. Actually, at that time, we use a peptide synthesis to obtain the amyloid domain because it's a pretty small amyloid domain. So since 2015, we have been working a lot on this system, and we now develop a new strategy which is based on the design of chimeric entity. So in this case, we design a single gene encoding for two prion-forming domains called R2 and R2. And we incorporated in the sequence an unstructured region between the two amyloid domains. And actually using this trick, I mean, that's a biochemical trick, we could recombinantly express and purify the chimeric R0, and surprisingly, the amyloid fold was quite robust. And this chimeric construct also behave as a prion in the fungal host. And it also gives high quality, very high quality solid-state NMR data. Actually, it also demonstrates that this chimeric construct, I mean, the way we design it, it didn't induce a local polymorphism. So we use this entity actually to solve the high resolution structure of R0 amyloid fibrils, again using solid-state NMR. So it forms a beta solenoid with a well-defined hydrophobic amyloid core. And so this approach to use a chimeric construct was indeed very efficient for us because we could produce recombinant R0 from E. coli and also label the protein with carbon-13 and nitrogen-15 isotope. And this would have been very expensive, not to say impossible to do, using solid phase synthesis of the short amyloid domain. So the approach was so efficient that we also used it to design a chimeric construct to study at high resolution the heterotypic interface between R0 from NWD2 and the R2 domain from HETES. So now we produce a single protein with a sequence that encodes the two amyloid domains, and again we put an unstructured loop between the two domains. Again, here it was quite fascinating to see that this heterotyping chimeric construct also behave as a prion in Podospora and Seminar, and we solve the structure of this R0, R2 construct using solid-state NMR. And here there's another spectroscopic advantage of our approach, is that we can actually derive the natural intermolecular interface between R0 and R2. So they come from two different proteins. That's because, and that's let's say the beauty of this trick. That's because the intermolecular interface is actually derived from the intramolecular interface between the two domains due to this sequence design. So it's actually much easier for us to determine intramolecular interface compared to intermolecular interface, because then we would need a much more sophisticated isotope labeling strategy to do it. So the big picture of this project is to be able to propose a model for amyloid-based templating mechanism. So in this model, NWD2, and so via its amyloid domain R0, would aggregate into beta solenoid and then recruit HETES and establish a specific heterotypic interface, so between R0 and R2, and this will trigger the assembly of HETES. So we're able to generate a high-resolution model of the complete fibrils. So using the high-resolution structure of HETES, the high-resolution structure of R0 we solve, and also the HETES NWD2 interface we also use using solid-state NMR. And you can see here this structural model of the NWD2 HETES fibrils. And actually, that's an approach we are currently investigating in the context of other functional amyloids, especially functional amyloids found in bacteria. Okay, so that's what I wanted to say today. So a few points I want to discuss as a conclusion. So we demonstrate the sin-free synthesis of amyloid proteins for application in NMR-based structural biology. Quite interestingly, we observe that for this system, so for HETES, we have a spontaneous aggregation in the cell-free reaction mixture. So we were able to obtain fibrils directly from the mixture without further denaturation or purification. And we show that this approach is very beneficial to introduce strategic isotope labels such as carbon-13 or also fluorines. For the second part, we demonstrate that atomic resolution structure of amyloid fibrils could be derived from proton-proton restraint using fast magic angle spinning. From a structural point of view, we established that it is not the amyloid fold per se that prevents the prion strand formation, but rather specific evolved sequence of the prions. And actually, that's also why we also designed this head and also this HEC synthetic prions. And we could also show that despite low or very low sequence identity, two prion homologs can adopt the same backbone structure, but surprisingly, they also do not cross-seed. And from, let's say, a more biological point of view, it suggests that natural prions, because these are natural prions, are evolutionarily polished to prevent structural plasticity and strain formation, because this structural polymorphism might compromise the advantageous seeding barrier. So there must be a subtle mechanism that is coded at the level of the sequence, also at the level of the three-dimensional fold, but as I say, not only at the level of the three-dimensional fold, that will allow or that will not allow the cross-seeding between two prion entities. And for the last topic, we develop an approach to get high-resolution structural information using solid-state NMR to derive the heterotypic interface between two functional amyloid domains. Good, and thank you very much.
Thank you very much, Antoine, for the excellent talk. While we are waiting for questions, please post your questions in the Q&A folder, or raise your hand. We'll enable you to join the panel. So Antoine, in your second topic, proton-based distance constraints, I think, and the FAST-MAS, do you exactly accurately measure the proton-proton distances, or you are assuming the short, medium, long range distances?
Though actually, so what we have done is we use a range. Actually,
Okay.
we didn't use the intensity. I mean, because we think that was not enough precise. So, you know, in solution NMR, it's possible to make different categories depending the intensity of the NOEs. Here we actually only use a range. I don't remember, but it's like from 2 angstrom to 6 angstrom. I don't remember exactly. I think just because right now it's still very difficult to use the intensity of the NMR peaks to derive more precise distance restraint. Yeah.
Great. Another technical question is in a F19 NMR, the 1D NMR spectrum you showed for isoleucine, I think, CF3, 6 fluorines, but you saw 5 peaks. Do you expect to resolve all the peaks, and why is that? Is it frozen or what?
So, I mean, there are 3 leucine in the hydrophobic core, so we also believe that they are rigid. So 3 times 2, it means we expect to have 6 peaks. I think probably due to some issues with the resolution, we think that 1 peak is actually, I mean, it's just too broad, and then 1 peak is just this additional peak is also in this range. I also want to mention that due to technical issue, we have recorded fluorine NMR without proton decoupling, just because on our probe we can or do fluorine detection or proton detection, but we cannot do both. So probably if you apply high power proton decoupling, we should get also better resolution and maybe see the 6 peaks if they are isolated and if they have different chemical shift.
So this is under 60 kilohertz or 100 kilohertz?
60 kilohertz, yeah. Yeah.
Okay, are there any other questions from the panel?
Yes, I have a. Can you see me?
Yeah.
No.
Yeah. We can.
Okay, I have just a comment for Antoine and a question for Hilal. The comment for Antoine is that sequence, we know that it is not so strictly related to structure. This is true not only for amyloids, but also for globular proteins. Just to present an example, ubiquitin and SUMO, they share only 15% of sequence similarity, but they are over impossible three-dimensional structures. So this is quite normal in nature. Next, I have a question for Hilal, and my question is HTT full length or fragments of the HTT protein a substrate of proteasome? In other words, is it known whether this protein is present as polyubiquitinated forms in inclusion bodies?
Lashuel, you are muted.
It is muted.
There is sufficient data to show that if you inhibit the proteasome, it results in increased accumulation of Huntington. You know, there is also data showing that Huntington inclusions are also ubiquitinated. Most of the work that has been done has been usually looking at the degradation of a very specific fragments rather than looking at whether the full length. I mean, there are data, although there are data also shows that if you inhibit the proteasome, you have slight increase of the full length protein. So it's very clear that I think Huntington degradation is regulated by both combination of autophagy and the proteasome system. But, which fragment and which form, this is, how they're differentially, degraded by the proteasome is not clear.
Thank you.
Hilal, do these fragments aggregate in the presence of full-length protein at all?
Yeah, I think this is what we tried to do in this experiment, right? We tried to generate, because most of the time they've been studied alone, and we tried to see if we generate them from the full length under controlled condition, but mimicking what would happen in the cell.
Yeah.
So now you have a mixture of full length, we have a mixture of fragment. To be fair, to be honest, under the condition we're using where the fragment is degraded rapidly, so we're actually moving now to try to do this in cells with controlled release of the different fragments, and to be, some sort of a spatial and temporal control to be able to validate this hypothesis in a cellular system. But we think they do because Huntington, we also think that Huntington in, the interaction with its natural substrate, binders like HAP40 could influence this process. So it could be that the ones that are not bound to their substrates are the ones that are more susceptible to proteolysis. We don't know because, as you saw from the cryo-EM structure, that part of the protein remains flexible despite in the complex and with the different polyQ. But in the cellular environment, it may still develop a different folded structure, or interact with different domains or with different protein. That's why I think the right way to test these ideas is now to take this concept into cells and neurons and test it.
Thank you very much. Magda, do you have any questions? They're muted. Magda, you're muted.
I have only kind of one to Hilal, more for clarification. The beautiful tomography pictures which you showed of the inclusions, are they? Can you kind of tell us again in what type of cells did you use?
So all the images of inclusions, as I showed, the cytoplasm. First, we looked at cytoplasmic and nuclear inclusions in HEK cells.
Oh, so it's okay. So
the first part was in HEK cells, and in HEK cells, because we wanted to study inclusions by the same protein in two different compartments in the same cell under the same conditions, right? And so those were actually not tomography; those data were correlative light electron microscopy. And the technique is usually, you use antibodies or fluorescent labeling to identify a specific cell or a specific inclusion. And then you're able to take that same cell or inclusion and look at it by EM to correlate it. The one where we showed tomography is looking at the video is looking at Huntington inclusions in neurons. The last part of the talk was nuclear inclusions in neurons. And I think this is, the previous images, as I showed, suggested some sort of a filamentous structure, but they were never very clear. But our data suggests, again, that's in the nuclear Huntington, they're predominantly fibrillar aggregates.
So these neurons, they were transfected with Huntington?
We use
one of your fragments.
We use lentiviral mediated overexpression of exon 1.
Okay.
All the cellular models I presented were with exon 1 Huntington.
All right. Okay. Yeah. Thank you so much. That's really helpful. Yeah, it's a beautiful data. It's really nice to see everything put together, because we as biochemists always like to just look at the things one at a time. And right now, when you put up everything, you can get more insights in what is actually going on.
Yeah, I think the complexity in terms of, the different fragments is something that is not unique to Huntington, because when Huntington, we think of different fragments. When it comes to other protein, you also have a mixture of different truncated forms. Truncation is a common modification for tau, synuclein, and PTMs. So the idea that it's a one protein, one mechanism of aggregation is probably not the case. I think that's, how do we deal with, modeling and understanding the complex interaction and aggregation of multiple homologs at the same time? I think this is something we're going to have to learn how to model and study, increase the complexity beyond sort of one disease, one protein, one mechanism. I think the second implications of our work is that, although the first step of going from monomer to fibrils is driven, can be likely reproduced in cells, it is clear now that what we produce in cells is different, from E. coli proteins is different from what's in the brain. Because all the cryo-EM structures of amyloid proteins isolated from the brain have very different structures from those we form from recombinant protein. And a lot of this has to do with two things: post-translational modifications and the complex environment where this aggregation process begins to happen. So what this means is we need to begin to take these complex systems and understand the grammar of aggregation again, so that we can refine our in vitro model systems, to be able to, to replicate the structures that we see. I think the final point is, maybe I want to emphasize again, there is much more to pathological aggregate than just the fibrils. The fibrils is just the first part of the process. And if you review our work at Lewy bodies, you can see there where we can see that these fibril structures are not end products. A lot of the PTMs we actually see in pathology happen after fibril formation. And they get significant remodeling, and they interact with other cellular component and lipid to form the inclusion. So the inclusions is a complex mixture of the aggregate plus everything, many other things that are in the cell. And I think if we began to study aggregation in terms of a process, not individual entities, we can begin to explain a lot of the discrepancies that we see between what we observe in vitro and what's in the human brain.
Yeah, I entirely agree with you. It's but this is of course quite challenging to account for all the components and stuff. But it's the way the thing should.
I always say that the answer is, by embracing complexity. We saw from Antoine's work in terms of cell-free system of generation of proteins. Our lab has been at the forefront of showing, chemical synthesis of proteins where we can reproduce the entire proteoform of synuclein, every single form that exists in the brain, blood, and CSF. We now have cellular models that allow us to see this complexity. So I think it is complex, but if we begin to embrace and approach that complexity, it's tractable. The other option is to stay doing what's simple, but it's not, doesn't recapture this process. The way I see it is we need to deconstruct this complexity, learn from it, and go back and improve the sort of our simple systems to be more relevant to the biological process. Because ultimately, we need these cell-free systems to understand things at the structure and sequence level. There are a lot of, there are a lot of things we cannot do in cells, we cannot do in vivo, but we need to bring these in vitro systems to be closer to what's happening in the brain in this case, in case of these diseases. And I think it will be possible.
No, that's really a great idea. Thank you.
So there are no other questions. Thank you both very much for great talks and great discussion. I really appreciate taking time out.
I think there was a question for Antoine.
Oh, there's one question here. Okay.
Aphrodite.
From Ada, a question to Antoine. Which are the earliest interaction mediating cross-seeding with your chimeric constructs? You are muted, Antoine.
Sorry. So it's a very good question. I'm not able to answer just because we look at the final state. which is the cross-seeded amyloids. When you know you already have a huge network of hydrogen bonds. So I mean, solid-state NMR is not the technique to answer this, to respond to this question, because we look at the final product that is already well formed. So we cannot say what are the key residue that maybe start to interact together before forming this very stable interface. I guess molecular modeling might maybe help to be able to analyze what are the first step of this event.
All right. So with that, thank you both very much.
Thank
And
you
we'll
for
close
having
this
us.
session.